welcoming all the colleagues and students and also the invited speakers for today's event. So we are having this event in honor of Professor Anju and her contributions to the field of chemistry and biology, and also to the institute during the more than 2 decades of being in the institute. So welcome all. I'll hand over the meeting to Professor Guhan so he can he can say a few words about Anju's contributions for this department and for the institute.
Hello, can you hear me? Yeah. Good morning and welcome to this mini symposium on chemical biology on the occasion of Professor Anju Chadha superannuation. So I don't know whether I'm the right person to give a keynote address. That's what it's listed, but I'll just make a few remarks. Chemistry has always been intertwined with biology, whether it's in the development of drugs or the unraveling of molecular mechanisms of a cell. For the last 50, 100 years, the chemistry has played a very strong role in biology, and that is undisputable. The fact that half the Nobels in chemistry are in the area of biosciences is perhaps proof of that. Or maybe it underscores the importance of biology. Anyway, I have known Professor Anju for the last 26, 27 years or so, even before either of us started our journey in IIT Madras. I think I first met her when she was working at the SPIC Pharma Limited, and I was at Anna University. Subsequently, she joined the chemistry department at IIT Madras, I think around 96 or 1997. And I joined the chemical engineering department a few years later. And both first started our journey in the Department of Biotechnology together. She joined the biotech during its inception. And perhaps both of us joined mainly because it gave us an opportunity to interface with biologists. The biotech department has always been an interdisciplinary department with a mix of traditional biologists and those working in the area of biomolecular sciences, either with a physics or a chemistry background. We have computational biologists and biological engineers. Of course, this diversity has always, has sometimes posed unique challenges, but I feel it gave us certain strengths, which also made us quite unique. So something about Professor Anju's work, although I'm not probably very qualified to describe that in detail, and Professor KKB will do that. So apart from some of the excellent work that she did in the area of organic synthesis, Professor Anju's work focused mainly on biotransformations and in the area of enzymatic catalysis. She also worked with some other faculty in the area of development of biosensors. So I'm very glad that we could organize this function in her honor, and I'm sure that some of the speakers, especially Professor KKB, would capture her contributions in much greater depth. It is especially heartening to see some of her colleagues from IIT Madras as well as PIC, Dr. George Thomas is here, give talks along with some of her students who have done exceedingly well and are now in prestigious academic positions. I'm sure you'll all enjoy this mini symposium. And with that, I would like to welcome Professor KKB to give the opening lecture. Thank you.
Thank you. Am I audible? Hello?
Yes, yes, you are.
Yes. Sir, can you wait for a minute?
Yes, please.
Yeah. So welcome to this mini symposium on chemical biology. So basically, I'm just moderating this technical session. Today we have 4 speakers for this session, and the first speaker is Professor K.K. Balasubramaniam. So we all know that he is a organic chemist in our country. So he has completed his PhD in 1965 from University of Madras. So later he carried out his postdoctoral research at the University of Madras and Wayne State University, Detroit, USA. And in 1971, 70-71, he came back to Department of Chemistry of Madras, worked as a scientific officer. Later he joined IIT Madras as a lecturer in 1971. So then he got promoted to assistant professor or associate professor, and then professor in 1990, and then 2001 he got retired from IIT Madras and took various positions in the industries, pharma industries like pharmaceuticals, Chennai. And later he joined our Department of Biotechnology, IIT Madras in 2015 as a INSA senior scientist. Currently he joined as adjunct faculty in Department of Chemistry, IIT Madras. His major research interests are synthetic organic chemistry, heterocyclic chemistry, carbohydrate chemistry, and medicinal chemistry, and so on. So to name a few of his awards and recognition, so which are quite very popular in our country, he is a fellow of Indian National Science of Academy and Indian Academy of Science, and he got several lifetime achievements in Chemical Research Society India and Chemical Conference in India in 2012, and he was a guest scientist at National Institute of Standards and Technology, Maryland. And he also got several fellowships like Humboldt Fellowships, and he was a visiting professor at University of Nantes and France. And he had carried out several projects from various funding agents in the country. So not only academic projects, he also collaborated with a lot of industries in the country, and he has developed a lot of process for various pharmaceutical industries. And he has published so far 176 papers in peer-reviewed international journals, and he has guided 37 PhD students, and he is currently handling a couple of projects in Department of Chemistry. So apart from that, he also did a lot of service to the education in the country. He delivered famous lectures in various institutes, and also he has delivered lectures in the very famous DST Inspire for chemistry students. So it's very prestigious for us to have him to give an inaugural lecture on this symposium, and especially he will highlight the contributions of Professor Anju and how he worked with us and how they developed the chemistry in IIT Madras. So I welcome Professor KKB to take over the first inaugural lecture.
Very good morning to one and all. Thank you, Professor Satya, for your detailed introduction. It's indeed a great pleasure for me and privilege to participate in the symposium conducted in honor of Professor Anju Chadha's contribution to the cause of education research on the superannuation. First of all, I must confess that I am neither a biochemist nor a chemical biologist. Yet, I've been having a long association with Professor Anju Chadha, and in the next 10 or 15 minutes, I would like to share with you my perception of Professor Anju Chadha as a collaborator, as a mentor, and as a teacher. Okay, I'm not really going to talk about my research accomplishments and so on. I'm not really competent to talk about that. I'll just start with how I met Professor Anju Chadha. Way back around 1983 or so, after he joined Speak Pharma, he dropped in the office one of the days with a sample and asked me whether if I can help him to get optical rotation. He came all the way from Marimal Nagar, and we had a very old manual semi-automatic polarimeter, I should say, under the German Intelligible grant. Okay, I was able to help her, and subsequently started visiting me for more such measurements and so on. At the same time, I was working on a project on the synthesis of triphenane-based sesquiterpenes. A research scholar from Speak Science Foundation, my name is Mr. Paini, was working as registrar registration. In that connection, I had to take the help of Professor Anju Chadha. I'm going to just show one or two slides now but what is the research? Can I have the slide, please? Can you project the slide?
Yeah, we could see the slide.
Yeah, these are linear triquinane. Skeleton is shown here. 3 cyclopentenes used linearly. A lot of terpenoid compounds occur in nature having this kind of a structural motif. And we planned a synthesis, a totally new synthesis for this class of compounds. And one of the reactions required a detailed investigation of the mechanism. This is where I had to approach Prof. Anju Chadha for her participation and help. Next slide, please. Next one. May I have the next slide? See, this project required understanding of a conversion of a vinyl ether to an aldehyde. And this aldehyde is a starting compound for our terpene synthesis. And we need to be very clear about the mechanism of this transformation. Whether this vinyl ether undergoes this transformation in a stepwise process or a concerted process. Reaction is done at room temperature using lithium perchlorate and diethyl ether, a very clean reaction, giving very good yield. Nevertheless, this understanding the mechanism was very crucial for the successful synthesis. And if it goes by a non-concerted process, namely breakage of the oxygen vinyl carbon bond, the product should be racemic. If the reaction goes through a concerted process, the optically active ether should give optically active product. And we didn't have the expertise of making this optically active ethers because it involved resolution using lipase. And I know Prof. Anju Chadha has been working as an expert in this area. She has done a lot of work in Germany on this kind of reactions. So I requested her, and she was, I should say, spontaneously agreed to help us in this. Next slide, please. Next one. Okay. Sorry, go back to the previous one. Okay, go back to the previous. Please go back to the. And the synthesis of this vinyl ether is not so simple. It has to start with optically active cyclopentanol, alcohol optically active. The optically active alcohol has to be dissolved from the ketone, has to be made from the ketone, and then the racemic alcohol has to be separated by lipase catalyst reaction to isolate the acetate, optically active acetate, and enantiomeric alcohol. All these were very challenging, and Professor Anju Chadha took up the problem, starting from the cyclopentanone reduction to the racemic alcohol, and making the vinyl ether resolution, and so on. Finally, we were able to show that this reaction proceeds by a non-concerted process, by breakage of the oxygen vinyl bond. Okay. When the work was completed, we submitted this to Journal of Organic Chemistry. One of the referees said it's a good piece of work, it should be published. Another referee said it's a good piece of work, but it does not go into JOC. The editor also agreed it should not go into JOC and rejected the paper. Then I said, why bother about it? Let us send it to Tetrahedron, which was also a very popular journal. But Professor Anju Chadha was, I should say, adamant. She said, no, how can they reject the work? I will take up the matter with the editor. And some few mails went and forth, back and forth. Finally, the editor conceded, and the paper got published. Why I'm saying this here is, Professor Anju Chadha has extraordinary determination. If she's convinced about certain thing, she will go all the way out to achieve. And this was an example where we were able to publish a good piece of work in a very good journal, ACS Journal, Journal of Organic Chemistry. After this, I should say, our laboratory, I was telling her about my plans as to what I'm going to do. Okay, I said I'm going to move to Sargent Pharmaceuticals as an executive director. I'm going to dabble into pharma process work. Later, if possible, I'll come back to IIT. So in 2012, I moved from Sargent Pharmaceuticals as executive director to two universities. One, Ramachandra University, Porur, other is Abdul Rahman University, Engineering Institute, as visiting professor. It was a 2-year term. It was over by 2014. Then I thought I would like to come back to academic work. Thought of applying for INSA senior scientist position. Since I am an elected fellow, I have the privilege of at least getting a few years of fellowship from INSA. When I told her that I am planning to apply for this, she asked me, where are you going to work? I said, I have no idea about it. If I approach chemistry department, they may agree. I'm not too sure. Then she said, why don't you come to the department? I will talk to my colleagues and so on. And she took the initiative. She talked to the head of department colleagues. And I was very fortunate, I should say. The Department of Biotechnology was kind of to forward my application. And I also got the fellowship. And I was provided with a very good office. And the fellowship lasted till 2019. It's a 5-year term. And I should say that this was mainly possible because the initiative was Anju Chadha and also the head of departments, Professor Mukesh Doble, subsequently Professor Arunakaran, and the entire faculty of biotech. Okay? They made my stay very comfortable and very pleasant. And this was one episode, I should say, where our research collaboration was fruitful and also helped me to come back to IIT Madras. Okay. After joining INSA scientists, I suggested to Professor Anju Chadha that why not we submit a project on ethambutol, in a biocatalysis route. Ethambutol is a TB drug, and it's a very popular drug, and it's a very simple molecule, very old one, still popular. No good synthesis there based on green chemistry. So immediately she jumped, it's a good idea, we can do a green chemistry-based proposal. And this proposal shown here, it starts with the racemic diol, and it has to be oxidized selectively to the 2-keto-butanol. One of the enantiomers of the racemic diol will be oxidized, the other will be unaffected. This keto alcohol has been converted into the ethambutol through a selective transaminase reaction followed by coupling dichloroethane. This is one of the processes that envisaged. And this required a biocatalyst work in the field of expertise of Professor Anju Chadha. Subsequent transformations can be done by any organic chemist. Next one, please. Next one. Next slide. An alternative proposal also given in the DST project. A chemical oxidation of the diol to give the 2-keto-butanol followed by oxide and normal reduction and separation of the amino alcohol to the required enantiomer either by lipase or by amylase. The project was sanctioned, and when we wrote the project, Professor Anju Chadha said she would like to ask for an IR, spectrophotometer. I said no, this work does not require an IR to that extent. The funding agency will not agree. He said, no, Professor KK, we leave it to me. Okay, I said, if so, I don't want to discourage you, go ahead. And the project was submitted, she went for the presentation. And initially there was reluctance to perhaps sanction this IR. As usual, Professor Anju Chadha was very persistent, and finally she got that instrument in the project. Okay, this was an example where her determination richly paid dividends. Okay. Now I will pass on to my perception of Professor Anju Chadha as a mentor, as a teacher, and as a research collaborator. Before that, maybe I would like to also take the liberty of indulging in making a few remarks about her personal traits. Anju Chadha's command over English is amazing. She is a fiery speaker. She is a speaker par excellence. One of the most sought after persons for talks on popular science. And I have attended meetings where she also participated. In meetings and discussions, the way she communicates the ideas and puts forward her arguments always gives the impression that she is very authoritative, commanding, and sharp. Truth is, she is candid and never to be on the politically correct side. And she never hesitates to call a spade a spade. Often because of this, her tough exterior demeanor, she is often misunderstood and criticized as being arrogant and disrespectful. But I have moved with her for more than 2.5 decades. I would say that I have found her to be extremely affable, honest to the core, helpful, humane, and ethical to the core. I have had the pleasure of teaching jointly with her the BTech and MSc. I found her to be extremely passionate about teaching and training students in the lab. She gave as much importance for teaching as she gave for research, unlike most of us who concentrate more on research and less on teaching. She used to come well prepared and was always punctual and encouraged students to come out with a lot of questions. And it was more of an interactive class that she always took. And I should say that in spite of the kind of impression that colleagues get about her, she has been a very successful research collaborator. And she could not have succeeded such a vast number of collaborations had she not been accommodative and accepting dissents and accepting others' arguments and so on. Okay? And she collaborated with various departments, inside IIT, outside department, also with industries and outside foreign universities. She has had collaboration with chemical engineers, electrical engineers, and chemists, and from industry on a wide range of topics. She is a very good example of a researcher who is able to work in several interdisciplinary areas and also be very productive and successful. It doesn't happen normally. We often take collaborative projects, but we never finally see the light of the day. This has been also my experience. Okay? Of course, her research expertise is very vast. Enzyme purification characterization, mechanism of enzyme reactions, assays, developing new biocatalysts and biocatalyst organic synthesis, organic molecules characterization, green chemistry, chiral chemistry, biosensors, enzyme-based, enzyme-free, bioproduction of value-added products, biodiesel, and whatnot. And no wonder that she has been recognized, okay, as outstanding women scientist and honored with several awards. To mention a few: Institute Chair Professorship, Women Extraordinary by Rotary Club, SK Chatterjee Award of Women Alumni for Excellence in Science, the International Women's Day Award for Outstanding Contributions in the Field of Science by University of Madras, and Elected Fellow of the Tamil Nadu Science Academy. Most important is her name figures in 100 women scientists featured in of this daughter's book, authored by the Indian Academy of Science. Okay. And because of her exposure to industry and also hands-on experience in industrial research, she has been highly successful in choosing research problems related to industry needs and society. It's just not purely academic work. If you look into her publications and her funding of projects, you will see a lot of industry-related projects in that. This has also been reflected in her mentorship. I have participated in many of her Saturday research group meetings, and I know the rigor with which these meetings are conducted. She never spared any student if she or she had not come prepared. He insisted on punctuality, honest reporting, recording, maintaining the lab notebooks, collecting the data, and full usage of instruments available in the lab. And she used to flare up, and you can see how the stones used to tremble. This is the exterior part of it. But when it comes to recommending the students for any fellowship, any job, and so on, you will also see the human side. She used to portray all the plus qualities of the students and try her best to always encourage and recommend. Okay. The presentation of the weekly research by the research scholars would look more like an American PhD defense, not a formal presentation at all. There's no formality there. And the students used to tremble until this meeting is over. No wonder that many of the students were grabbed with prestigious universities like University of Hyderabad and for faculty positions, and also by industries. In fact, two of our research scholars were able to start their own industries as soon as they graduated, mainly because of the mentorship of Professor Anju Chadha. Okay? I'm waiting for the day when our national science academies will also elect her for the fellowship which she deserves. Okay? It's also sad to, and I should say that I personally feel that despite her tremendous achievements in science, as a speaker, as a popular science writer, and as a mentor, okay, she has not been given due recognition in her own institute, I should say. I would expect her to be made automatically IIT Madras scholarship I really don't know the basis of which she was not able to take up the position. But sometimes, system always has its flaws. Like my case, I took 20 years to get my promotion, whereas others all my colleagues got in 6 or 7 years. So this happens in the system. Perhaps you can't blame it now. But anyhow, I'm very happy that the students of Prof. Anju Chadha had taken the initiative to conduct this wonderful seminar to honor her on her occasion of the Superannuation Day. I would wish her more years of active personal life and good health. And I must say, I would continue to cherish my association with her. Okay? I still look forward to doing some collaborative research with her, wherever she is going to be it Punjab or Jammu, it doesn't matter. She is one who always jumps at any kind of new ideas and starts planning to do immediately the work. Okay? I hope the new ideas would make use of rich experience in research, teaching, and mentoring. Thank you all. Have a good day.
Thank you, Professor. Thank you, Professor, for very interesting and motivated speak about your collaboration with Professor Anju Chadha in Department of Biotechnology. Now, is Jerina is there? You said she want to play some video.
Hello, dear Anju, dear friend and colleague. First, let me congratulate you to your retirement party this week. Although I'm not sure whether such a congratulation will be welcome, it will be, as we say in Germany, a goodbye with a laughing and a weeping eye. I'm now 15 years emeritus, and I'm still missing my laboratory and the teaching of young scientists. I always hated setting exams and especially marking them. Maybe it's the same for you. Nevertheless, it was so nice to have met you as a Humboldt Fellow in my lab almost 30 years ago, and I very much enjoyed your wonderful hospitality during my frequent visits at the IITM. I hope to see you again if Corona allows, and wish you for the future all the best, especially good health and time to pursue still some science, I hope, and also many non-scientific activities you may have. All the best, and stay healthy, and I hope to see you again, your friend Manfred Schneider.
Okay, thank you, students, for a nice video. So we'll move on to next lecture. It's by Dr. George Thomas. So Dr. George Thomas did his MSc Biochemistry from PGI Chandigarh, and he did his PhD from IIC Bangalore in 1984. Then immediately after his PhD, he moved to National Eye Institute, NIH, Calcutta, USA. He did that postdoc for 5 years. Then he came back to India and joined SPIC Science Foundation. And he worked there for 14 years at various levels in SPIC Science Foundation. In 2003, he moved from SPIC Science Foundation to Interfield Laboratories, Kochi. There he was working as a lab chief. In 2017, he moved to Chief Operating Officer, Agri Genomes Labs, Kochi, and currently is working there. And to his major contributions, so it's quite important to tell to all young people in the country. So he was involved with the first gene cloning in India, where he cloned the histone gene cluster of rice, and also involved in the first DNA sequence in India, where part of histone gene cluster was sequenced, and studied a lot of transcription of histone genes of rice. And he was involved in the first ever plant histone genes to be studied. So as a result of this, he got first Nature paper in his PhD work. And he's also involved a lot of genome sequencing of various plants. Currently, his research activities are involved in the tilling CRISPR-Cas9 based genome editing and expression of several proteins, important proteins in plants. So as a part of his scientific work, he got several recognitions. He got best thesis in biological sciences during his PhD time. And he got Best Life Science paper published in Indian Journal, and he was a member in DPT task force on biocontrol agents, and he was a member of various several committees like Fulbright Fellowship Selection Committee and faculty selection committees in various universities, and he was also involved in the Rockefeller Career Development Fellowships. So to his credit, he has produced 7 PhDs when he was working at Spitz Science Foundation. And it's our pleasure to have him in this symposium. And I request George Thomas to give his lecture on journey from genes to genomes. Please, sir.
Okay. Yeah, can you hear me?
Yes, sir.
Yeah, good. Yeah, thank you very much for inviting me. It's a pleasure to always talk at an occasion honoring my dear friend Anju Chadha. So I'm going to talk about my journey from genes to genomes, and you'll also see that Anju was always with me in my journey, although at a distance, but we were always traveling in the same direction. So I joined Indian Institute of Science for PhD in 1977. It took me 7 years to do the PhD. First 3 years, I was groping around for finding a decent problem. And the next 3 years, I finished my work. And the last year was spent waiting for the degree because NIH needed PhD degree before I could go for a postdoc.
We finished together. That's fine.
Yeah. Okay. So basically what I did was, as said in the introduction, we made a genomic library of rice. And that is quite an achievement because we didn't have any facilities. Please remember, we didn't have Google, we didn't have any radioactive material that can be imported. We didn't have proper restriction enzymes. Nothing. We just had PBR322, one of the most primitive plasmids available. And sometimes we used to get gifts of enzymes from our seniors who had gone to the US. In any case, we made a very primitive genomic library, and we could identify a fragment, a clone, which carried 3 histone genes of rice. And that is the biggest, the luckiest event, I would say, because it's like a needle in a haystack. Now, how did I identify the histone genes? That's a very circuitous way of doing things. It's not like doing a blast analysis today. I had to capture messenger RNA, the messenger for histones, using my clone, then elute the messenger, do an in vitro translation. That in vitro translation was done using a rice embryo translation system that we developed in the lab. And then identify the protein. So we identified, and it is quite a lot of work and enough for a PhD and more. But one question popped in my mind. I have 3 histone genes, H2A, H2B, H4, in a fragment of 6.64 kilobases. Now, this DNA fragment has 2 strands. What bothered me was whether these 3 genes are encoded by the same strand or they are distributed over the 2 strands. And I simply could not sleep. And I couldn't write my thesis until I answered this question. So it's very difficult. At that time, we didn't have any of these facilities. Spent several weeks in the library trying to find out a way of doing it. And suddenly one paper came up which described how you can separate strands and isolate and purify them, with simple agar electrophoresis. So I don't know if you can see the figure on the right. So this is the, I'm spending a little time on this because I think this is my most intellectually satisfying research that I did throughout my career. And this particular picture that you see, it is taken from my journal, my Nature paper. That is the biggest or the happiest moment in my life perhaps was seeing this gel. Now this is a strand separation analysis. This is the double-stranded DNA. Can you see my cursor? I don't know if you can see. So the lane 2 shows the double-stranded DNA. Lane 1 shows where I tried to separate the strands. And this agarose electrophoresis was done inside a fridge, a refrigerator, for 2 days, 48 hours, I think, at very low voltage. And luckily, the strands got separated. So this is about 60% of the DNA got strand separated, 30% remained double-stranded. Of course, then I analyzed it, again identified the different proteins and found that H2A and H2B is encoded by one strand, and H4 is encoded by another, the complementary strand. And this came in Nature because it was a very significant finding at that time. After that, I moved to NIH. Okay, before that, Indian Institute of Science is where I first came across Anju Chadha. And as people have described about her nature and character, it is not something that developed later in life. I think even in the institute, she was a very strong personality. She would speak her mind. She would call a spade, be it her fellow students or her professors, or even the director of the institute. So I moved to National Eye Institute after getting the degree, because I wasted 1 year doing that, and worked on crystalline genes. So all of this time I am describing when I worked with genes, individual genes. So histone genes and now crystalline genes. Crystallines are the eye lens proteins that give transparency to the lens. Very interesting work, but I'm not going to spend too much time on this. The significant thing is that I get to NIH, there are a few Indians there. Some of them I didn't know, some of them I knew already from the institute. And who was there among those people I know? Anju Chadha. Then I moved to Spic Science Foundation because I felt the need to come back and work in India. My education was supported by National Science Talent Scholarship, and I felt obligated that I should come back to India. So I came back, joined Science Foundation, set up a biotechnology lab, and produced 7 PhDs. And so several genes were studied, individual genes, mostly isolated from rice, and one of them, arotinate, was codon optimized for plant expression and synthesized in my lab. And each of these is a story that will take half an hour to explain, but I'm not going into all that. So I worked on individual genes and their promoters, but also during that time, what happened is Spic Pharma was set up by Dr. M.D. Nair, who turned out, eventually became a very good friend of mine. He lives in Kochi and we interact very regularly. So who should he recruit other than Dr. Anju Chadha? So from IISc to NIH, sister department, same campus. And now we came to Chennai, where we were in sister companies, but of course, geographically a little bit separated. She was in Maramalai Nagar. But our lab was on her commute route, and so occasionally she used to drop in to say hello. And that went, developed into a collaboration with my colleague, Dr. Lokeshwari, who used to run a tissue culture lab, where a compound, she was synthesizing a compound, and it was always giving a mixture, racemic mixture. So she wanted an enantiomeric excess, purity. And so that was done by using carrot cell cultures. And this was published, I think it was in Tetrahedron. So that is one collaboration that my department or my center had with Anju Chadha. And by around 2000, Spic started declining and they were closing down Spic Science Foundation. I think Anju sensed it much before and moved to IIT before calamity struck, but I was foolish enough to stay back and face the calamity. Anyway, I moved to Interfield Laboratories. So here I was involved in some food testing, nothing to do with genes except for testing some pathogens. And there was an interesting situation where, the shrimps are cultured with some antibiotics at time, to protect them from infection. And these antibiotics should not be there in the shrimp that we export. So the European Union decided that these four are AOZ, AMOZ, AHT, and semicarbazide are the markers to look for certain nitrofuran metabolites. Nitrofurans, when they are consumed by the shrimp or by the humans, get converted to metabolites which are very toxic apparently. But the European Union scientists themselves said SEM is not a good marker, but the bureaucrats wouldn't care for that. They set SEM as a marker. Now in 2010 or something, I think 39 or so shipments of shrimp were rejected from Brussels, saying that they contain SEM. But from the same factories, shrimps that were sent to Germany were not rejected. So we had done some work in the beginning stages of setting up this analysis, and we knew what was happening. I went about proving it clearly. We knew that the shell of the shrimp, scampi, which is the large shrimp that you get, freshwater shrimp. The shell of the scampi had some SEM, which is inherently present. It's naturally present. It's not coming from antibiotics. So we found that in Germany, they homogenize the material with the meat alone, whereas in Brussels, they homogenize it with the shell. And we knew that was the problem. So I collected freshwater shrimp that was never exposed to antibiotics from Tapti River in Gujarat. I personally went to Chilka Lake in Odisha, and also from Vembanad Lake in Kerala. And extensively studied this and showed that in shrimp that is not exposed to antibiotics, there is SEM present inherently in the shell. We sent it off to European Union through our diplomatic channels, and they got shocked. So, but they were very open. They commissioned a project through University of Ghent to study this. And they came out with a report saying that this is indeed true, that in Brussels, the method of sample preparation is the problem. And in other European countries, it will not be detected falsely like this. So in a way, European Union will probably be more cautious before blaming India or maligning India. So after that, in 2010, while I was still working in the field, I became an advisor to Science Genome Group, which was set up by some friends and my junior colleague from Speak Science Foundation in Cochin. And they were requesting me to join them. And so eventually in 2017, I joined as CEO of AgriGenome. Now, so far I've been working with genes, and suddenly a paradigm shift has occurred in the early 2000s when human genome sequence was announced, and subsequently it changed completely. You can get an entire genome sequence. At that time, it used to take about 2 weeks, but today you can do it in maybe 2 days. So the entire genome can be sequenced, not just one gene. And so that paradigm shift was very useful, and so we went about doing a few genomes in our group. The first one was the first report on an Indian female whole genome sequence, and this is that paper that came. The first male sequence was reported by Vinod Skaria from IGIB. Ours was the first female sequence that was published. Around this time, a few sequences were done all over the world, and then some consortia were developed in the US and in UK and all, to do a global study on whole genome, because the main application is in personalized medicine, and for that you need basic information, the genomic information. So such consortia were working, but Indian samples were not represented. I think they had a Telugu person's sample from Houston, and a Tamil person's sample from UK, but samples from India were not going. I don't know. I think there were some restrictions on sending out samples from India. So we got excluded from all of these studies substantially. So our company, group of companies decided that this has to be solved. And then, you can do a typical Indian genome, but what is a typical Indian genome? India is populated by people of different ethnic backgrounds, different genetic backgrounds. You can't have one single gene that represents all of India. So we decided that we'll do a large number of genomes, and eventually it was also expanded to all of Asia. So that is called the Genome Asia 100K project. That is 1 lakh genome, so that it will give us a very good baseline data about the genome diversity. And the first results came, was published in Nature in December last year. They also thought it fit to, feature it on the cover of Nature. So we were always doing work on wildlife genomics in our company. We studied many diseases of the wild elephants, the wild monkeys, and we were also studying snake. And one thing that bothered us for several years was the snake bite, the complications, death and other even worse than death complications that arise out of snake bite. So how can we address it? And all, we had a few symposia organized, and finally we decided to take the plunge and do the whole genome sequence of the Indian cobra. There are 4 venomous snakes in India. The first one we did a very high quality, that's what is known as a reference genome sequence was done. We identified about 129 potential toxin genes. That means genes that can produce potential toxins. And out of this, 19 were found to be expressed in the venom gland of the cobra. And all the sequences, the entire sequences, and you can see that this is an open access journal. So, it is freely available. The sequences are freely available. Anybody can make use of the data, produced in this paper and develop antivenom using various different strategies. It's not very easy though, because there are 19, and so you need to produce 19 antibodies, and it's a little difficult, but at least we opened the door for such possibilities. So this appeared in Nature Genetics on the cover this year in January. We did I've always been working on rice. So, I was very interested in this purple colored rice called purple puttu, which all parts of this rice is purple in color. The leaves, the husk, the endosperm, everything, the grain, everything is purple in color. And my friend, Dr. Arjula Reddy, you will see his picture a little later, worked on this for more than a decade. And so when I was asked to choose a rice for whole genome sequencing, I said purple puttu. We wanted to understand, and we had some very interesting results in this paper on, we were questioning the common belief that a certain deletion of 14 base pair fragment was responsible for the color phenotype, and we have now questioned it in this paper. So it is an important paper. We did the whole genome sequencing of a virus. This is a virus that infects a tea pest. And so we are now trying to develop a biopesticide, which is an insecticide that is not toxic. So we're trying to develop that in collaboration with the Tea Research Association. We were interested in doing whole genomes of many plants. We thought that plants of interest to India should be the focus. So what better than medicinal plants and spices? The first one we chose was curry leaf. I don't have to explain the importance of curry leaf. I think everybody uses it in the cuisine. And it has also, I'll tell you in a minute. So this is the whole genome of curry leaf. A very high quality genome has been developed. The paper the manuscript has been written. It will be submitted this maybe within 2 weeks or something. So this is a very significant achievement. So while this was being discussed in my lab, they talked about some muriaquinin or some such molecule that is present in curry leaf, which led me to think, oh, this hydroxyquinone is I may be wrong in terms of chemistry, so pardon me, but my basic thinking was like this. Hydroxyquinone is a repurposed drug for COVID. So can any of these secondary metabolites in curry leaf have anything to do with binding to COVID proteins? So I told my people to have a look at that. They did some studies and then they extended the studies, and they looked at 69 secondary metabolites and their in silico interaction with COVID proteins, all the different proteins. I think about 7-8 proteins were looked at. And we found that about 6 secondary metabolites we could identify which were binding to the COVID proteins better than the currently used repurposed drugs like remdesivir or any of those drugs. So this paper has now been accepted. It should come out in maybe 10 days time. I think the proof was submitted on Thursday. So in about 10 days, you'll be seeing this out. You can have a look at it and see if you can make your own decision whether it will do any good to you. It's only an in silico study, no experimental proof. But I have started consuming mint and lime and orange and cocoa and what else? Curry leaf and all of these things which have important, and tea. All of these have important secondary metabolites that possibly might help you in preventing. If the viral titer in your nostril is low, maybe this will prevent it from getting worse. Anyway, so we are currently working on many other genomes. This is the cardamom, and we are also doing phyllanthus. Phyllanthus, as you might know, is a very good Ayurvedic medicine used for hepatitis, and also Krishna Tulasi. These two are in collaboration with other labs. We did some work. This is not genetic transformation. This is just plain mutagenesis. It's called tilling. So we got a rice line that gives us a 15-day advantage. So this line is already ready for harvest, whereas this is the control is only starting to flower. We got another line that gives about 20 to 25% extra yield. So this we have now, filed for registration with PPVFRA, that is the Protection of Plant Varieties and Farmers' Right Authority. For if we have to release it, we have to get their approval. So it has been submitted. It will take a couple of years to bring it out. So now coming to the CRISPR-Cas9, we have gone one full circle, started with individual genes, went on to whole genomes, and now I'm back to individual genes where by editing or mutating certain genes can we achieve some goals. So this, of course, as you know, got the Nobel Prize this year for the work done by Jennifer Doudna and Emmanuel Charpentier. The question was whether we can demonstrate, because before jumping into a major project, we should show that we are capable of doing it. So can we have a visual scoring method to prove our ability? So this is the pathway of the carotenoid biosynthesis. All trans lycopene, which is the red lycopene, is produced from the precursor is tetracis lycopene, which is tangerine in color or orange in color. Can we knock off this gene by CRISPR-Cas9 and produce orange colored tomatoes rather than red colored tomatoes? To cut a long story short, we did get the orange colored tomatoes after this editing event. And you can see it a little better in this picture. You see the color difference. So we looked at the genes of this plant, the mutant plant, and clearly we could see that there are mutations. Whatever we intended was there. So it has worked at the molecular level, at the genomic level. But what happens? What is the situation with lycopene? In this symposium, if I don't show one HPLC chromatogram, it will not be fair. So we did the HPLC analysis. These are 3 different wild type fruits, and this is from 3 different mutant lines. You can see that there is a sharp big peak of lycopene in all the wild type, which almost disappears in the tangerine colored mutant lines. So I think this proves that we have been fairly successful, and we now can reasonably be sure of doing CRISPR-Cas9 editing for bigger projects. The other thing that where Anju and I collaborated or worked together was in conducting the Joseph Thomas Memorial Lectures. As you know, Dr. Joseph Thomas was instrumental in setting up the Biotechnology department in IIT Madras, and he passed away, I think, in 2007. So we started memorial lecture series at IIT, and the IIT, whoever was the head of the department and Anju Chadha have been very, very helpful in getting this organized. So we had some excellent speakers. As you can see, G. Patnabhan was the first one to give the lecture, followed by D. Balasubramaniam he was from CCMB, but in LB Prasad Institute now. Professor Dharmalingam from MKU, Professor Balaram from IISc. And this Balaram talk was the one where Anju had to really run around, organizing things because his flight got delayed due to, earlier flights in Delhi being delayed due to fog. So from Bangalore to Cochin, he could not, Bangalore to Chennai, he could not come. And then within a couple of hours, we organized a satellite link, and we were able to give something like this webinar where he spoke and it was displayed on the screen. Because some 200, 250 students had come, and we just could not cancel it like that. So that was the most tense organization of the JT Memorial. Then followed by Arjula Reddy. I talked about him a little earlier. He is the one who worked on Purple Puttu. Professor Sharad Chandra, he doesn't need any introduction. Dr. Nagaraja from IISc, Deepankar also from IISc, Dr. Lalji Singh, unfortunately he is no more with us. Followed by Tangaraj, was also from Lalji's group. And then we had Dr. Gadakar talking about all the insect studies that he did, the behavioral studies, and Usha Vijayaraghavan, the plant biologist from IISc. So in this lecture series also, Anju helped me quite a lot, and I'm hoping that we can continue to have these lectures in IIT with the help of the chairman, the head of the department, and other faculty members. And that's all I have to say. Anju has been a great friend, and whenever I come to Madras, she makes sure that we have a meal together, and it's always a great pleasure to be speaking in a symposium that is honoring her. Thank you very much, and Anju, good luck. We'll keep our contacts and keep meeting. Thank you.
Thank you, Dr. George Thomas, for sharing your wonderful journey from gene to genome. It's quite inspiring for young scientists who can also look into identifying various genomes which are of medical interest. Students, do you have anything to play?
Hello, Anju. Hello, everyone. I have known Professor Anju Chadha for almost several projects. We have had joint proposals, we have published together, and we also had research students. She is quite a versatile person. She had collaborations from faculty, from chemical engineering, civil engineering, electrical, chemistry. She always wanted to give her best in whatever she was doing, and she expected the same from others as well. So there was no shortcuts. She is quite versatile because I was surprised. She was learning Sanskrit a few years back and also preparing for the exam. She was very friendly with the people around, especially those who were in the cleaning staff. We have had long discussions over a cup of tea on topics ranging from structural zinc, catalytic zinc, or the selectivity of candida, or even where do you get the best Bakar Vedi in Pune or best Maharashtrian food in Chennai. So I wish both Anju and Ramesh a happy and healthy and satisfying career ahead. Thank you.
Okay, thank you. So next speaker is Dr. Deepti Sarin. Deepti Sarin did her MSc Biochemistry and PhD from MTech Chandigarh in the area of microbial biotechnology in 2002. Later, she moved to postdoctoral research at University of California, San Diego for 2 years. Then she moved back to India and joined as a young scientist DST CRC at CSAR AMPRA Bhopal. And then she joined Department of Biochemistry, Punjab University as assistant professor. And currently she is an associate professor there in Department of Biochemistry, Punjab University. And her major area of research interest are identification of mutative genes by genome mining, heterologous expression of various proteins, gene knockout techniques, and protein and peptide purifications. And she had completed 4 projects from various funding agencies of 2 crore rupees. And currently she is working on success relationship studies of novel 2-component antibiotic rosunin. And she has published nearly 17 papers and she got 1 US and an Indian patents. Okay, and she also contributed several book chapters in Springer publishers. So now I request Dr. Deepti Sarin to give her a lecture.
For honoring Professor Anju Chadha, who has been a great friend, though we have a huge age gap, but we are, I don't know why, but we have a great association. And for quite many years now, I don't remember exactly, maybe 8 to 9 years. And we just happened to meet in some ways, we do have some common interests. Biotransformation is maybe one of them. And then I also am, I have been associated with Dr. Padi, and I would also thank Dr. Padi to have, who has just come for my rescue and for the illustrative talk that he has given. I would straight away come to my talk before I say anything else. We've been working on enzymes. To start with, I worked on biotransformation. And then my approach is a little bit different. Instead of going for regular screening of enzymes through soil screening, I happen to do it through bioinformatics. And so that's why I give my title as mining. So through genome mining approach, we have digged some industrial enzymes for biotransformation, and my interest was in epoxide hydrolase. I said was because I'll explain why. Also in non-ribosomal peptides, and now I am more into ribosomally synthesized and post-translationally modified peptides, which are actually many. There are 18 different peptides known. They are all different classes. I am mainly into lentibiotics and thiopeptides. So I would just like to give an overview into what I am interested in. So this is a very old paper. We did some bioresolution of racemic phenylglycidyl ether by a putative recombinant epoxide hydrolase. And this was taken from Streptomyces graeceus, NBRC. This is the strain number. So what we did was, from a racemic mixture of styrene oxide, we did kinetic resolution by a recombinant EH, and we could obtain R1-phenylethane 1,2-diol. Actually, many epoxides like 1s2R indene oxide, ethyl 3,4 epoxybutyrate, and many others, They act as chiral synthones for drugs like HIV protease inhibitors, neuromediators, non-steroidal anti-inflammatory drugs, neuroprotective agents, and beta-adrenergic blockers respectively. So that's why we are interested in, because they act as chiral synthones. And they are actually alpha-beta hydrolase family members. And they being enantioselective selective enzymes, they are, I just said, they are employed to kinetically resolve racemic epoxides into enantiopure But for the kinetic resolution of various epoxides, microbial EHs are being explored because of their ease of large-scale production. And recombinant whole cells offer many advantages over the purified enzymes as they require less cumbersome preparation with no downstream processing steps, thus saving both time and resources, and can also be immobilized to be reused through various numerous cycles. Now, our approach is bioinformatic analysis of putative EHs. So this is due to, as I think Dr. Thomas explained, due to the advancement in bioinformatics, a significant progress has been made in the discovery of novel biocatalysts. This became possible due to the massive amount of information on microbial whole genome sequences deposited in the public databases such as NCBI, Tiger, Sanger, etc., made available. Now, most of the enzymes annotated in these genome databases are not functionally confirmed, so they represent new treasures of uncharacterized biocatalysts. So what we do is we screened the genomes of the two organisms. One is Candidatus pelagibacter ubique and another is Tachybranchia nauseensis for the presence of putative epoxide hydrolases. And we actually looked for the conserved residues which are present at the active site and conserved motifs which are already published and known to have been playing a role at the active site. And they are known as, let me show you the next slide. So here, so we did a blast search using known EH sequence as a query. And here is the structure of the alpha-beta hydrolase family EH or epoxide hydrolase. We scanned for the specific motifs and the catalytic triad residues, and we also did cluster W alignment of the putative EHs with the known EHs. So to name them, they are like HGXP motifs and SXMP motifs, and also the catalytic triad are specific residues like aspartate, two aspartates which are distant, and a histidine. So to name them, there are two motifs and three residues, which is like very few residues, but amazingly, this is a very conserved kind of residues which are present in all the epoxide hydrolases, but we have to scan them carefully because lipases, nitrilases, and other examples of alpha beta hydrolases have to be subtracted from this class because their catalytic triad residues are a bit different. So it was a very tricky job to actually come down to strongly epoxide hydrolase, or strongly we can say what I can say something which is very promising, but it was a very tricky job. So finally, we could actually characterize 3 epoxide hydrolases, though we failed in 3. So it was a like 50-50% success. So we could actually fish out 6 of them. 3 of them were successfully characterized and 3 of them did not come out to be successful epoxide hydrolases. So it was a very tough kind of a call. So that's why right now I'm not pursuing this project. But anyhow, right now, so this paper actually gave us a molecule, a protein, which could give us all these products. So these many molecules were found to be positive against it, like these many phenylglycerol ethers, thioglycerol ether, chlorophenylglycerol ether. So all these substrates it was able to catalyze with this much of retention time. So these are the methodologies that I'm displaying. And here is the enantiomeric axis and the E values. I learned a lot with Dr. Chimney in Department of Chemistry at GNDU, and that's how I came in touch with Dr. Anju during this project, which was DBT funded. But since it was a very tricky job, so I did not pursue it further. And again, this tells the biotransformation where we got how much is the yield. So this was 40% yield, 98% EE with S-propane diol. And so this is the comparison with the already existing epoxide hydrolases in the literature. Then slowly we moved out of this project and we went into non-ribosomally synthesized peptides. And so this is the modular nature of the NRPS. I'm more interested in this because this is more of a genomic mining project. Now, what are non-ribosomal peptide synthetase? Now, NRPS and PKS, which is polyketide synthases, they are present in bacteria and fungi, and they are major multimodular enzyme complexes which synthesize secondary metabolites like the pharmacologically important antibiotics and siderophores. Each of the multiple modules of NRPS, which consists of adenylation, condensation, and thiolation domain, they activate a different amino or aryl acid to synthesize a linear or a cyclic natural product. Like we see here, there are each of the modules are named as 1, 2, 3 up till 6, and the 6th module has a thioesterase. The first module does not have a condensation domain. So this is just an example, which is a very worked out example of actinomycin. So the studies on NRPS domains, the knowledge of their gene cluster architecture and tailoring enzymes have helped in the in silico genetic screening of the ever expanding sequential or sequence microbial genomic data for the identification of novel NRPKS. So what we were doing were, we were looking for novel NRPKS and PKS clusters. And our approach was to look for these novel clusters through the adenylation domain. Now, adenylation domain is an integral part of the NRPS and is the substrate selecting unit, because each of these substrate which is being tagged here, and so that's how the final product is made, is selected by this adenylation domain. Now, it being the integral part, the number of NRPS modules and their sequence corresponds to the number of amino acyl or aryl residues in the NRP, representing a collinearity rule. The A domain, the A domain is responsible for the substrate selection and activation. So the activation is shown here, which is then transferred to the thiolation domain. So here is the activation with the help of ATP, which is then made an AMP adduct. The thiolation domain contains actually a phosphopentethene arm, which swings between the adenylation and condensation. So here is the pentethene arm, which swings between the adenylation and condensation and acts as a carrier of the amino acyl, or where is the cursor? Okay, amino acyl or the aryl thioester residue, and is hence responsible for the extension of the product. The C domain catalyzes this catalyzes the peptide bond formation right here. So we did a review. First of all, we understood the literature and we did a review in the Indian Journal of Biosciences, and this was reviewed by Dr. Debashish Mohanty, who is a known bioinformatic person in India, and we were blessed. And we learned a lot doing this review. And also we had gained a lot of confidence in the bioinformatic tools during this time. So the process of activation and condensation is repeated until the last module, which contains the additional TE, which I said thioesterase domain at the C terminus, at which the chain terminates. So as I said about the tools, so these are the tools which we used: NCBI Genome Viewer, NCBI ORF Finder, BP-ROM, TMHMM, CDD analysis, MSA code softwares, and the other softwares which I've given in table 1. I took this table, this figure from the same paper. Now, once these complete clusters are predicted with approximate product chemistry, which we did judge in this paper, I'm going to show you just a glimpse of the clusters. Then the product has to be actually characterized in vitro by physicochemical methods, because its properties can be studied by in vitro reconstitution approach, gene inactivation, comparative metabolic profiling approach, heterologous gene expression, comparative metabolic profiling approach. So these are the clusters that we actually, these are the novel ones, which we actually fished out from Stachybrandtia nauseensis. This is our favorite culture somehow. So these are some of the, clusters. This is PKS and NRPS hybrid cluster. We can see AT, CAT domains, and here is a KS as well, and methyltransferase as well. And this is again a hybrid cluster we can see. And we don't know what the product is. One of the very amazing findings that we got in that paper, as a proof of concept, we also published in this review only some novel clusters. This is MBTH homolog kind of a gene, which is found in every PKS NRPS cluster, which was not known in many publications earlier. But these days, what I am now realizing that in every paper, MBTH gene, whose function is also not very well explained, but somehow maybe it explains that, some people explain that it helps in folding or the substrate binding of the A domain. So that's what it does. It's a very small protein, MBTH. So we also covered this MBTH homolog in this review. So this is cluster 3 and cluster 4. Again, most of them are hybrid. Well, this is one of the clusters that we characterized to some level, and we also explained the approximate product chemistry. That is, which one is the substrate that each of these A domains picks up, and what is the final product that it lands with. So these are some of the publications in this area in epoxide hydrolase, biotransformations, and non-ribosomal peptides. And then we moved on to the ribosomally synthesized peptides using all these tools again. And we got interested in this because those were the days when we were getting some, publications about the AMR, antimicrobial resistance. A lot of reports about that. So we thought, and more of, those times we were also learning that these days we are getting funding only on translational research. So we tried to look for something which can land us into a product, good product. So this is something that that actually led to a product which was antimicrobial. So we looked for clusters, again using those tools, and we published a paper titled Novel Lanti-Associated Lentibiotic Clusters. So this is a cluster which is actually a lentibiotic cluster. It codes for 2 precursor peptides, a lentibiotic synthetase, a protease, and a transporter encoding gene, immunity proteins, and a response regulator. And this is coming from this cluster, or BGC, biosynthetic gene cluster, is coming from Streptomyces roseosporus, NRRL. Again, this is all from genome. Once we identify the clusters or BGCs from any genome, then we get hold of the culture from a respective culture collection. So as a proof of concept, we did the heterologous expression of this roseosin, which is a 2-component lentibiotic, and we developed a novel product which is directed against drug-resistant pathogens. Now, these lentibiotics are actually antibiotics, and they act against antibiotic resistant, for example, methicillin resistant Staph aureus and vancomycin resistant cocci. They also target biofilm forming pathogens. And so they offer tailored solutions to specific problems. So our achievement is we overexpressed roseosin. Roseosin is a formulation which is actually 2 peptide mixed formulation. So these are the advantages, that if we have a high producer of lentibiotic after heterologous expression of antimicrobial peptides, it is a solution against the chemical synthesis, because in chemical production, the stereochemically inaccurate lenthanin rings is an issue. Now, what are lenthanin rings? I'll be explaining a little further. Okay, let me go to this slide first of all. Now, as I said, these are ribosomally synthesized peptides. So there are 2 kinds of residues in these peptides. This is enriched in serine and threonine as well as cysteine. We all are chemists here. Now, serine and threonine, together with cysteine, they are able to form, first of all, they get dehydrated to form didehydroalanine and didehydrobutyrate, respectively, serine and threonine. And then after Michael addition by the cysteine residue, they form lenthanin or beta-methyl lenthanin ring. And this is what is post-translational modification. And this post-translational modification is done by actually an enzyme, which is encoded by this synthetase, lentibiotic synthetase, or ROSM. We call it ROSM. And since this is coming from Roseosporus, so we've named it ROSM, ROS1, ROS2, and ROSM, and so on. So after getting post-translationally modified in vivo, the peptide is structurally constrained and is not able to get cleaved by the protease, which is encoded by within the same BGC. So let me come back. How does it act on the cell to act as an antimicrobial? So this is how. Here is the membrane of the bacteria. I don't know why is it going further. Here is the membrane structure of the bacteria. This is the lipid 2 precursor. Now, lipid 2 precursor has this diphosphate moiety. This diphosphate moiety is actually bound by niacin, which is a very well-known antimicrobial compound used worldwide as a food additive. And once it is bound by this, it forms a pore with lipid 2 and itself, and hence the cell becomes leaky. As a result, there is an antimicrobial action. Now, this is a single component antimicrobial or lentibiotic, while in our case, roseosin, similarly to lacticin. So lacticin and roseosin are similar in only one aspect. Lacticin is also developed by another lab in US. They are also, this one is also a 2-component lentibiotic, alpha and beta component, same as in our case. Our lentibiotic is a bit different. I'll be showing in the next slide. Here, alpha component actually encapsulates the lipid 2 precursor first. After making a complex with the lipid 2, beta component comes and makes a whole. So that's it. Now, 2-component lentibiotic contains alpha to anchor with the pyrophosphate group and beta component to cause membrane permeabilization, and hence this makes it a stronger complex, and the efficacy is increased with the very less amount of lentibiotic required to do the same action as that of niacin. So, this figure actually, the cluster W alignment shows how is our different. So here is the lacticin. The roseosin actually has a longer peptide with more cysteine residues and hence more lenthanin rings and hence more constrained structure. And we found that its MIC is quite low. MIC is minimum inhibitory concentration. So this is actually the technology behind the roseosin production. So we are doing in vivo modification, which is lenthanin rings formation or post-translational modification in vivo, purification and production in vivo. After that, we are cutting the N-terminal leader peptide in vitro, and then the synergistic action we are testing in vitro. So here is a bit of characterization where we are doing the characterization by MSMS Malditov, where we found number of dehydration, number of lengthening rings in alpha is, well, M-ROSE1 is actually beta because it comes first in the BGC, so it is A1, but actually this acts as a beta component. It comes later and binds to the complex. So A1 has 6 lengthening rings and A2, which is alpha, has 4 lengthening rings, and we have found that it also has 1 disulfide bond. Okay, so this was very interesting work. We are also now trying to find the structure, decipher the structure through NMR, but we have not been successful as yet, but we are trying hard. Yeah, this shows that the synergistic activity, we have already published it in molecular microbiology. If we add the 2 peptides in 2 wells, we only get an oval zone of inhibition. Otherwise, whenever antimicrobial compound is added, we get a circular zone of inhibition. So only when the 2 are added together, wherever they meet, they diffuse together, there is a zone of inhibition. Individually, they do not give any zone of inhibition. So here are the MIC levels compared to niacin, which is available commercially. It is comparable. Yeah, and so these are the publications this year and earlier. The patent is already filed on this work. Yeah, recently, this work also got recognized through the Carbex CCamp AMR Quest 2020. And this led me to attend an accelerator program where I got a lot of headache, in the sense there's a long road to go. If I want to develop it into antimicrobial, there is a lot of work still to be done. So, okay, so I need to acknowledge the funding agencies who have helped me setting up my lab and maintaining it, some glimpse of my lab facility, and my earlier students who are already through. And I have currently 4 students working. They are not shown here. And here is my department where I work, and I've been serving here for last 15 years. Yeah, and I thank you all for your patience. And thank you, Dr. Anju Chadha, for being my mentor always.
Thank you very much, Dr. Deepti, for your excellent lecture on genome mining and antibiotics. So thank you very much. Now we move on to next lecture by Santosh Kumar Padi. So I'm happy to introduce him because he's our own student of Department of Biotechnology and did a PhD with Professor Anju Chadha. Basically, he did a MSc from Berhampur University, and he was a topper there. And then he joined IIT Madras for PhD position, and then he completed PhD in 2005. And then later he moved to postdoctoral training at various places. He did first postdoc at University of Minnesota. Then he moved to so quite difficult name. And Smart is
it's Greifswald
University, Germany. And then he joined as a faculty at BITS Pilani in 2012. Later he moved to University of Hyderabad, Department of Biochemistry, School of Life Sciences as assistant professor. So he has published more than 20 international peer-reviewed papers, and currently he's working there. And I request Dr. Santosh to give his talk.
Yeah, am I audible?
Yeah.
Good morning, all. It's my privilege to talk about our work in the occasion of the superannuation of Professor Chadha, who is my mentor. So I'll talk about a small piece of work, a one-pot enzyme cascade catalyzed conversion of primary alcohols into chiral beta-nitro alcohols. To begin with, let me tell you the importance of the chiral beta-nitro alcohols. So in this slide, you see two boxes, each having a number of structures. And as you can see, the top one shows top one shows a number of drug molecules with S configuration, and the bottom one shows a number of drug molecules with R configuration, R chiral center in them. And both these kind of molecules can be synthesized from amino alcohols, 1,2 amino alcohols, as you can see here. Chiral amino alcohols, if you look at the enzymatic way of the synthesis, then they can be synthesized from a cyanohydrine. Cyanohydrine can be synthesized by addition of hydrogen cyanide, which is a toxic molecule, into aldehyde using hydroxynitrile lyase. This is an enzyme. On the other hand, they can also be synthesized by simple reduction of beta-nitro alcohols. Beta-nitro alcohols can be prepared by addition of nitromethane into aldehyde. However, nature has not evolved a nitroaldolase to carry out such reaction. So we looked at the different biocatalytic routes for the synthesis of chiral beta-nitro alcohols. As you can see here, the most common one is lipase catalyzed kinetic resolution. A kinetic resolution is often limited. It's always limited with maximum 50% yield. And we looked at the literature, all the methods known to prepare chiral beta-nitro alcohols using kinetic resolution, and they take very long times, from 12 hours to 7 days. In addition to that, they also need a deprotection step sometimes. They can also be synthesized by enantioselective epoxide ring opening using an enzyme called halohydrine dehalogenase, but again, that is a case of kinetic resolution. To circumvent this, dynamic kinetic resolution have come up. However, dynamic kinetic resolution uses not complete biocatalytic, it uses a chemical catalyst to racemize and then use a biocatalyst carry out kinetic resolution. And the reported method uses takes about 20 days, which is quite long. Asymmetric reduction of prochiral carbonyl compound is a popular method. However, the carbonyl compounds need to be prepared here because they are not readily available. These are alpha nitrocarbonyls, and they are not readily available. Other method is a retro-Henry approach, where a racemic Henry product can be cleaved selectively by hydroxynitrile lyase, abbreviated as HNL. Again, this is a case of kinetic resolution. This is an efficient method, that is a nitromethane addition into aldehyde by Henry reaction. In fact, this is an atom economy process. However, Henry reaction, which is catalyzed by a few hydroxynitrile lyases, as I say, abbreviated by HNL, is often limited with substrate inhibition, which means when you carry out the reaction at higher substrate concentration, the substrate itself inhibits the enzyme. So as I said, the enzyme hydroxynitrile lyase, let me introduce what a hydroxynitrile lyase is and what it does in nature. Hydroxynitrile lyase catalyzes as it is a lyase, it catalyzes the cleavage or lysis of cyanohydrine into hydrogen cyanide and a carbonyl. And these alpha hydroxynitriles are appeared in a biological system by the breakdown of cyanogen glycosides, which are kind of secondary metabolites in higher plants. But in a one or a more step process, beta glycosidases cleave the sugar moieties and they produce alpha hydroxynitrile lyase, and that's how the hydroxynitrile lyase cleaves the cyanohydrine into aldehyde and a carbonyl and a cyanohydrine. However, in laboratory, these enzymes are used for the synthetic purpose, where hydrogen cyanide is added to a carbonyl to produce optically pure cyanohydrines. A few of them have shown so-called promiscuous property, where they add nitromethane instead of, apart from HCN, they also add nitromethane into a carbonyl by producing a optically pure beta-nitro alcohol, which is the aim of this project. So we looked at all the methods known so far, especially the hydroxynitrile lyase known to carry out the enantioselective addition of nitromethane into a carbonyl compound. The first one was reported in 2011, almost 10 years ago. And this enzyme which catalyzes this reaction is ATHNL, abbreviated as ATHNL. It is a hydroxynitrolase from Arabidopsis thaliana. However, it is limited with 30% conversion, as you can see, poor yield and moderate enantiomer selectivity. Only 91% enantiomeric excess was there, and also with substrate inhibition. 5 years later, one more group carried out similar work, and they reported the enantiomer selective nitroaldol reaction by two other enzymes or two other hydroxynitrolases. One is ACHNL, another is GTHNL. And these two enzymes also showed very poor enantiomer selectivity and conversion. And they did protein engineering, and they could found a mutant which could improve the conversion and the enantiomeric excess. And in 24 hours, this reaction produced in good conversion and yield. We, in this project, targeted to prepare these compounds, not by protein engineering, but by simply adding another enzyme into it. As the major problem in this nitroaldol reaction is the substrate inhibition, which means aldehydes are very reactive. I mean, aldehydes are reactive compared to their reduced counterparts like alcohols. So we thought starting with alcohol and introducing an alcohol dehydrogenase, like a horse alcohol dehydrogenase in situ, could produce benzaldehyde or the aromatic aldehydes, so that the aromatic aldehydes will not be accumulated, and that can be treated with ATHNL in situ to produce beta nitroalcohols. And we were successful, as I will tell you how we proceeded. So first we characterized the ATHNL, the Arabidopsis thaliana hydroxynitrolase. As you can see, the extreme left one is an SDS page, and there are the various steps of purification. The last one shows the pure protein. And then before using the enzyme in biocatalysis, we have to characterize it kinetically, and we used one of its common substrate, mendylonitrile, because mendylonitrile cleavage is a very common assay to carry out to characterize a hydroxynitrolase. It is very clear that the enzyme had showed good Km and Vmax values. Subsequently, we carried out the enzyme kinetics, and the Michaelis-Menten plot is evident here. As you can see, the Vmax and Km for this promiscuous reaction, and these are the Km and Vmax values for the racemic mendylonitrile and racemic NP. We compared with the literature report, and we were pretty good with the enzyme, and then the enzyme is ready to do biocatalysis. The next enzyme is HLA-DH, or horse lever alcohol dehydrogenase. We also purified it and characterized. This is the SDS page, as you can see. But as we are ready with the two enzymes to proceed to carry out the enzyme cascade, we had to look at the various inhibitions that are possible. For example, in this case, we use two enzymes, and we have to look at the compatibility of the enzymes with the substrates. The first thing we looked at is the benzyl alcohol inhibition on ATHNL. This benzyl alcohol, as it is a new substrate for the ATHNL, we wanted to find out its inhibitory effects. As you can see here, up to 50 millimolar substrate concentration, the specific activity has minimally reduced. That indicates that benzyl alcohol can be used in this enzyme cascade without much problem, as up to 50 millimolar, not much inhibition is seen. Then we tried to understand the inhibitory effect of benzaldehyde on HLA-DH. So benzaldehyde is the product by HLA-DH, but nevertheless, we wanted to study by increasing the amount of benzaldehyde. And as you can see, we studied up to 200 millimolars of benzaldehyde. With increasing benzaldehyde concentration, the activity decreases, but not really much. And as you can see, the Ki and the Km values, the Ki value is 0.01 millimolar and the Km value is 0.007 millimolar, indicating that the enzyme has higher affinity for the substrate, for the benzyl alcohol, than the benzaldehyde, which is an inhibitor, which means Km is much less than Ki. Therefore, this can be used in a cascade without much problem. And of course, we also studied the effect of benzaldehyde on ATHNL as predicted. It was seen that with increasing benzaldehyde concentration, ATHNL activity drastically decreased. And we characterized this first time. It appears like this is a mixed inhibition. When you say mixed inhibition, that means the substrate could bind to the enzyme or the enzyme substrate complex. The inhibitor could bind to the enzyme or to the ES complex. So both the possibilities are there. We have characterized the Ki and the K small I and the K big I. K small I indicates the inhibitory constant of enzyme to the inhibitor, and that is 33.9 millimolar, but K capital I is 3.5 millimolar, which means the inhibitor has higher affinity to the enzyme substrate complex. We are yet to find out how this is done at the molecular level. Nevertheless, this is a good piece of information. Having found that the enzyme cascade can be carried out compatibly without any inhibition, we then moved to carry out the enzyme cascade. The first hurdle in front of us was the compatibility of the two enzymes with respect to the pH. HLA-DH is compatible or has higher optimum pH optima 8.8, and ATHNL has pH optima 6. And to understand this, we have to understand the mechanism of HLA-DH. HLA-DH is a dimeric enzyme where a zinc present in its catalytic site plays very crucial role in its catalysis. I've taken these pictures from the corresponding references, as this is a case of enzyme without substrate. It's an apo-enzyme. As you can see, zinc is present here and water is over here. Zinc has made a tetra coordination here. And here the water is replaced by the alkoxide ion, which is subsequently oxidized by NAD+. And the predicted mechanism is given by in this paper, Biochemistry 2004. So this clearly shows that zinc plays a very important role in the oxidation of alcohol into this, the primary alcohol into the aldehyde. And what happens to zinc at lower pH? This is a very important piece of information that we learned from a paper which published almost 50 years back. And it says that when you decrease the pH and carry out the HLA-DH catalysis, which means the oxidation of a primary alcohol, the specific activity decreases. As you can see here, with the decreasing pH, the specific activity decreases. Well, not only the specific activity, also the zinc content decreases. So that makes sense that with decreasing zinc content, the specific activity decreases. But at the same time, the paper gave us a solution that by supplementing a little bit of zinc, as you can see here, the specific activity could be retained. This is without zinc addition and this is with zinc addition. As you can see, this is at pH 5 and pH 5.5, and the activity could be retained actually. So therefore, we proceeded by adding 0.1 millimolar zinc chloride into this biocatalysis, and the HLH, the horse liver alcohol dehydrogenase, worked perfectly in this. Now, as this pH issue coming up, we try to optimize several parameters which might influence the biocatalysis or the biocatalytic cascade. And to start with, we used different buffers and different pH buffers. As you can see, we used KPB buffer, potassium phosphate buffer, and also acetate buffer. Acetate buffer is a good one to catalyze horse liver alcohol dehydrogenase oxidation. And as you can see, in the case of acetate buffer pH 6, which is the last one, okay, let me clarify here. What I'm showing here is the blue one in the bars. The blue one indicates the conversion of benzyl alcohol to NP. This is popularly called as NP, and the nitrophenyl ethanol. And the orange one is the conversion of benzyl alcohol to benzaldehyde, which means up to this much, it is the conversion of benzyl alcohol to benzaldehyde, and the blue part is the conversion of NPE. So as you can see, with increasing, in the case of acetate buffer, the aldehyde content is very much. However, that shows that HLA-DH is working perfectly in acetate buffer. However, the enantiomeric excess, which is represented by the black dots, the enantiomeric excess is decreased. That means in acetate buffer, ATHNL is not working very well. So we have to consider the compatibility of both, and considering both these things, we have selected KPB-PH 6, which means potassium phosphate buffer pH 6 to start with. Then we optimized to cofactor recycling agents. As you can see in this reaction, NAD plus is getting reduced to NADH. It has to be recycled. That's where another carbonyl compound such as acetaldehyde or acetone comes into picture. This is going to, using this, we could convert back the NADH into NAD plus, which will be used subsequently in the reactions. Between acetaldehyde and acetone, acetaldehyde gave much better result, as you can see, and therefore we proceeded with acetaldehyde. We then studied the effect of ATHNL content from, as you can see in the previous slide, but in the case of acetaldehyde, the aldehyde content has increased, which means you have a lot of aldehyde, but then the aldehyde need to be converted into corresponding NPE, and that's where the ATHNL need to be optimized. And therefore, we optimized the content of ATHNL, and as can be seen, from 400 to 1,000 units of ATHNL were taken in 800 units. The content of NPE is maximum. After that, it is not much increase in the NPE content. It's the NPE concentration is there. Therefore, we have taken 800 units as the optimum value and then proceeded. Then we optimized the concentration of NAD plus, which is the cofactor. As you can see, the cofactor concentration with increase, when it is increased beyond 0.8, so this is 0.2, 0.5, and 0.8. Beyond 0.8, there's a decrease in the aldehyde concentration was noticed, and this is probably when you have a lot of aldehyde, the NAD plus, it's getting converted into NADH. The aldehyde, a lot of aldehyde produced, the aldehyde is also inhibiting HLA-DH, which we have seen in our inhibition studies. So we took 0.8 millimolar of NAD plus as the optimum value, and then we proceeded to optimize the HLA-DH content starting from 1 to 5 units. As you can see, from 1 to 5 after 2 units, the NPE content was almost same. In fact, the aldehyde concentration started decreasing. Therefore, we took about 2 units of HLA-DH as the maximum or as the optimum value. Subsequently, we moved to optimize the nitromethane concentration. Nitromethane concentration was optimum at 1.5 molar. As we understand that we need to provide excess of nitromethane to carry out the nitroaldol reaction. And at very low concentration of nitromethane, the NPE concentration was very low, which is obvious. And then we moved to optimize the organic solvent. Organic solvent plays a crucial role in this, usually in hydroxynitrolase biocatalysis, because organic solvents help in increasing the enantiomeric excess by minimizing the product degradation, number one, and also there is a product partitioning happens in organic solvent because we carry out the reaction in a biophagic solvent. So as soon as the product is formed, it moves into the organic solvent. Therefore, its degradation could be minimized. That's how the enantiomeric excess is retained. And among all this, we found good enantiomeric excess in N-butyl acetate. NBA stands for N-butyl acetate, and this is in coincidence with the previous. There are a few other hydroxynitrolases. We also saw good enantiomeric excess in N-butyl acetate. We then varied the concentration, or the content of N-butyl acetate from 45 to 65. And in about 50% N-butyl acetate, we found the optimum value of enantiomeric excess in the conversion, and that's how we proceeded with 50% of N-butyl acetate. Then we optimized the substrate concentration. In this case, we have not, this picture is not showing as the conversion, because with increasing substrate concentration, the conversion would decrease, even though the amount of product is increased. For example, when you say we take 40 millimolar of substrate, even though the conversion is less, there could be more amount of product. Therefore, a better way of representing is to calculate the number of micromoles of product formed. So we did that starting from 15 millimolar to 40 millimolar. As we can see, in 25 millimolar, we get good conversion. At the same time, enantiomeric excess, which is close to 98%. And beyond that, the enantiomeric excess started decreasing, actually. So from this study, we observed that we could increase the conversion from as little as 30 to almost 60%. However, we cannot completely convert all the aldehydes. Probably the aldehyde is still inhibiting, or to circumvent that, we thought of studying the effect of different stabilizers. In our previous study, in another study in our laboratory, we used sorbitol and sucrose, which are the polyols known to stabilize the protein structure. And we used these two, and we found in the case of sorbitol, there is an increase in the aldehyde content actually. And finally, the acetaldehyde, which is also known as the cofactor recycling agent, and its concentration was varied, and we found in about 125 millimolar, the aldehyde content was much more. Beyond that, it was not effective. And here is a time course study, which clearly indicates that with increase in time, you can see the decrease in the benzyl alcohol, which is the starting material, and this is the benzaldehyde. As you can see, the benzaldehyde is reducing with increase in time, and this is the percentage of NP, which is the final product. It's also increasing, and the percentage of enantiomeric excess remains quite high. So up to 4 hour, the enantiomeric, the conversion was increasing, and beyond that, it was not that much effective. Having optimized all the conditions or several biocatalytic parameters, we have. So this is the overall picture of how the different optimized parameters could increase the conversion and the enantiomeric excess of this enzyme cascade. You can see that there is a very big increase in the conversion by several biocatalytic parameters. We then tried to carry out this biocatalysis, this cascade biocatalysis with the different substrates, and the enantiomeric excess was quite good in all these cases, as you can see. However, the conversion was limited because the conversion was limited not only not because of the NP, but also because of the HLA-DS. The substrate has to be compatible. Nevertheless, we could find that the conversion was very good, almost 64% in the case of benzaldehyde, compared to 30% conversion by a previous report where they use only the nitro-aldol reaction. And this is with respect to the cascade. This is a HPLC chromatogram to show you that this is the standards. This is benzaldehyde, benzyl alcohol, and the R and S beta nitro alcohols. And this is a control. And this is the biocatalysis. After biocatalysis, the HPLC profile. As you can see, the benzyl alcohol is very less. There is a little bit of benzaldehyde, and it is only R product. S product is almost not there. This is for another substrate. I'm skipping the rest due to lack of time. So to summarize, I would say that we have designed and developed a new enzyme cascade for a one-pot synthesis of enantio-enriched beta-nitro alcohols. Our inhibition studies revealed that aldehyde has a mixed inhibition on Arabidopsis thaliana hydrocytolase, which could be a crucial information to explore the catalytic mechanism. It avoids the use of reactive aldehydes and uses inexpensive and easily available and stable alcohols as substrates. We used almost 10 different we prepared almost 10 different optically pure R-beta-nitro alcohols in up to 99% enantiomeric excess and 64% conversion using 25 millimolar substrate concentration, which is a decent one. We could find almost nearly or more than twofold increase in the productivity of this promiscuous nitro-aldol product compared to the existing HNL catalyzer synthesis. I would not say this is a complete study. There is still scope to improve the yield and yield. Probably hydroxynitrilase engineering could improve the yield and the substrate scope and enantio selectivity. And if somehow we can minimize the substrate inhibition further, probably this may increase the yield of the cascade. With this, I would like to acknowledge my team. This work was mainly carried out by Anju Chadha, and thank you all for your presence.
So thank you, Santosh, for a very interesting lecture, and wish you a good future.
Thank you, sir.
Yeah, thank you.
Hello, Professor Anju Chadha. Congratulations on your retirement. This is Romas Koslauskas from the Biochemistry Department at the University of Minnesota. Congratulations on your accomplishments and best wishes for a wonderful retirement. It's been a delight to meet you several times at conferences and also to work with the excellent students that you've trained. I know Santosh Kumar Padi was a postdoc in my lab for several years, as was Titu Devamani. Both of them had trained and gotten their PhD in your lab. More recently, Debayan Chaudhuri spent a summer in my lab doing some experiments. He's now a graduate student at the University of Wisconsin. I think you should be very proud of the careful work that you've done on the carbonyl reductases and careful enantioselectivity and applications to synthesis. And I've always admired your work, and it's a delight that you've reached retirement, and congratulations once again.
Rumas Kozlowskis, Minnesota, US.
IIT Madras, and I'm extremely happy to say these few words with regard to Professor Anju Chadha and this mini virtual symposium which has been arranged at the time of her retirement. She had joined this institute way back in 1998 with a rich experience of 8 years from Big Pharma. And I remember distinctly in the month of October 1998 when she had joined this department. It was an initial struggle because she has moved from extremely good labs at the pharma industry to the place where the labs have to be created from scratch. But however, she continued with grit and she did extremely well and later moved on to the Department of Biotechnology in the year 2004 and later rose to the full rank of professor in 2006. It was nice to know that she was an alumni of Pune University, from where even myself rose, and our interaction started fairly nicely, and during the process I learned that she had special interests at the interface of biology and chemistry, and that's the reason why she moved on to biosciences department. Her research interests have been always focused around biocatalysis, or to say enzyme catalyzed organic synthesis, and she has definitely made valuable contributions to the same. Her academic excellence and wide interest in industrial as well as academic objectives has enabled her to be in the national committees of eminence, such as the Progress Advisory Committee of DST and also Women's Postdoc Committee. She was also in the task force meant for the nutraceuticals under the institution of DBT. And I can confidently say one aspect, that she has been extremely forthright at expressing her views. She's extremely candid and she doesn't hesitate to call spade a spade. And I think this is a very rare quality these days. Professor Anju Chadha has evolved as a very eminent scientist, can be easily seen when she's featured as one of the scientists in 100 scientists in the book entitled Leelavati's Daughters, which was brought out by Indian Academy of Sciences in the year 2009. That is myself and my wife, Dr. Jaimala, who is also an alumni from Pune University, wishes Professor Anju Chadha a very happy, healthy, and peaceful retired life. Thank you for giving this few minutes in this retirement function. We wish everyone the same. Thank you.
Before we move on to the next session, I want to thank you, Professor Gumadi, for taking care of the technical session. I now request Professor Guhan Jayaraman to felicitate Professor Anju Chadha.
Thank you very much, Sneha. Are you able to see me and hear me clearly?
Yes.
Professor Anju Chadha joined the services of IIT Madras as an assistant professor in the Department of Chemistry on 26 October 1998. She was appointed an associate professor on 12th January 2004 and appointed as professor on 19th May 2006. Earlier, she had worked as a senior scientist with SPIC Pharmaceuticals R&D Center for 8 years. In 2004, when the Department of Biotechnology was started at IIT Madras, she was one of its early faculty members and has worked there ever since. She was made an institute chair professor in 2018. Professor Anju Chadha did her PhD from the Department of Organic Chemistry in the University of Science from 1979 to 1984. Prior to that, she taught at Fergusson College, Pune, after her master's degree in chemistry from the University of Pune in 1977. Professor Chadda's research interests are in the areas at the interface of biology and chemistry. Her work focuses in the area of biocatalyzed organic synthesis, especially asymmetric synthesis, enzyme mechanisms, development of enzyme assays, green chemistry, biodiesel, chiral chemistry and analysis, enzyme-based and enzyme-free biosensors. She has authored more than 100 original research papers as well as many invited reviews and is an inventor in over 8 patents and has mentored a couple of startup companies. Professor Anju Chadha has delivered several keynote addresses and invited lectures in various international and national conferences during her tenure. At IIT Madras, she has also been a faculty member of the National Center for Catalysis Research, Department of Chemistry, and the Center for NEMS and Nanophotonics, Department of Electrical Engineering, and has collaborated with the faculty of the departments of Chemical Engineering and Electrical Engineering because of the interdisciplinary nature of her work. At the national level, Professor Anju Chadha has been a member of the board of some national institutes, a member of the Program Advisory Committee, Chemical Sciences of DST, DST Women's Forum PDF Committee in Chemical Sciences, the Task Force on Food and Nutraceuticals of DBT, and various selection and evaluation committees. She has also reviewed projects internationally, namely the Swiss Science Foundation. Professor Anju Chadha is a recipient of many awards. The Hanumanta Rao Gold Medal for the Best PhD Thesis in Science Faculty of IISc Bangalore in 1984, the Fogarty Fellowship, NIH USA, 1985 to 88, the prestigious Alexander von Humboldt Fellowship in 1992-93. The International Women's Day Award for Outstanding Contributions in the Field of Science by the University of Madras. Also, the Professor S.K. Chatterjee Award, Indians of Science, given to a woman alumni for excellence in science in 2016. Professor Chadha is one of the 100 women scientists featured in Lilavati's Daughters, a book brought out by the Indian Academy of Sciences, Bangalore. She won the Woman Extraordinary Award given by the Rotary Club of Chennai and is an elected fellow of the Madras Science Foundation in 2006. Her work at IIT Madras includes more than 30 sponsored research projects in the capacity of a principal coordinator or investigator, and she continues to be a consultant for some of them. She has mentored 20 plus PhD students, 8 MS students, 5 MTech, 9 MSc, and 10 BTech students from biotechnology, chemistry, and chemical engineering departments and electrical engineering departments during their dissertations. The faculty, staff, and students of IIT Madras, and particularly the Department of Biotechnology and Chemistry, wish Professor Anju Chadha a long, happy, and healthy, and prosperous future and success in all her endeavors. Thank you.
Thank you, Professor Guhan.
So we have a small, I don't know, I'm trying to get back to my Zoom. Just 1 second, please. All right, I'm not. If you're, am I visible?
Yes, sir.
Okay.
Yes, sir.
Yeah. So this is the felicitation memento which we have prepared for Professor Anju. Are you able to see this?
Yes, sir.
I would like to
Let me take
virtually.
it a little back.
Okay, I'm very sorry. Yeah.
Yeah.
And go on a little down also.
Okay. Yeah. Thank you. Because I'm not able to see on the Zoom. Somewhere it disappeared. Are you able to see it now?
Move a little back, Guhan.
Okay.
Just a little back.
Okay.
So that you can see the lift slightly above.
Santosh, can you please spotlight Guhan? That way it won't keep switching. So
I would like to gratefully acknowledge the contributions of Professor Anju Chadha in the Department of Biotechnology and Departments of Chemistry, and wish her all the best for the future. And this is the plaque which I would virtually like to hand over, Professor Anju, and I'm sure we'll give it to your students in a day or two, and they will hand it over to you. Thank you very much.
thank you, sir.
Anju, you're mute if you're saying something.
Yeah, thank you, Professor Guhan.
Okay, thank you.
We'll move on. Now it
Thank you, Guhan. I just got the memento. Thanks a lot.
Okay, sure. No, we'll hand it over soon. Thank you.
Okay, so it is time to hear some more experiences ma'am's friends and students have to share with her.
Hello, this is Inakshi Bhattacharya from Electrical Engineering. Anju has been my friend and a collaborator for many years, even before she joined IIT Madras. When we were students, biology and mathematics separated fairly early in school, and never the twain shall meet types. So I had not studied much biology, and chemistry, though I had to study till I think intermediate, I was not very fond of the subject. And when I started working on sensors, there were all these interesting biosensors, and I knew a lot of things could be done with them, especially the miniaturized one. And I used to discuss them sometimes with Anju when she was still with SPIC. But we didn't start any work on that. But when she joined IIT, first the chemistry department and then later in biotechnology, we had collaboration over many years with several students, and so I did learn a lot of biology from, I don't know how much I learned. I learned enough to understand what was going on, and that was all thanks to Anju. Not only that, all the students who were joint students, MS and PhD, were all from electrical, and they also learned. And even though sometime during this course they would cry that we have to do too much chemistry or we have to do too much bio, finally they all did it and they enjoyed doing it. So I think it was very I don't know if I can call it successful, but it was very satisfying collaboration. And Anju is of course a very good teacher. All of you know that. I just remembered that during an evening walk in 2003, when the Chemistry Nobel Prize was communication on the cell membrane, just on that half an hour walk or so, she had explained it so beautifully. I think later she gave a talk also in IIT on that, but it was so, such a good combination of electrochemistry, electrical signal, chemical signal, everything. So, yeah, I'm going to miss Anju. We haven't been collaborating much recently, but my learning from the things I learned from my contact with her will stand me in good stead. So good luck, Anju, for the new innings, and I'm sure you will be as active as ever. Bye.
Dr. Ganesh Pandyan, Junior Associate Professor from Kyoto University, Japan, wants to share his memories about ma'am.
Hello, everyone. Can you hear me?
Yes.
Yeah. Okay. So my name is Ganesh. So I'm a proud Anju Chadha product. I can call myself like that. Madam used to call me Lucky Bug, if Madam remember. So I'm always been, Madam Trillian, thanks to you. First of all, very happy to see you after a very long time. And thank you, the organizer, for this opportunity to express my thanks to my guru, who inspired most of us by leading with example. And I think I'm one of the senior member of Anju Chadha lab. And we always had a very great rapport. And although I did not do my PhD, but my pre-doctoral studies is the main reason for me getting a position here in Kyoto University. The reason is because Anju Madam has been kind of a visionary. Even in 2004, when interdisciplinary study was not so popular, she had me and involved in so many interdisciplinary projects. And so Anju Madam is my first boss, and she opened the research gate for me. And how I joined is after appearing my final exam for my master, I came to meet Anju Madam. Anju Madam don't know me, and just by seeing me, I don't know, she encouraged me very nicely and asked me if I'm interested in doing some project. And when I started a project in her lab, my chemistry skills were terrible because I'm a biotechnologist. But ma'am quickly identified that I have a knack towards the biology, and she encouraged me to work on the biological aspect. And I was able to start the biocatalyst, Candida parapsilosis. And later, we went on to work on Candida species. And basically, I was kind of a project assistant who was maintaining the bugs. So that's why she calls me Lucky Bug. And she made me to involve in many industry aspects also, like Malady Pharmaceuticals, PIC, and Kelvin Chemicals. So everyone knows about Madam's magnetic personality and warmth and charm. But when I was unwell, one time I brought my mother, and Madam was so nice to my mother, and my mother was saying so many good things about her after meeting her. So she's not only a wonderful personality, but also a very nice human being. And so Madam's my time in Madam's lab opened not only my academic life. So I also met my wife, Dr. Vaijayanti, there in Madam's lab, and we are now here settled in Japan. So currently I'm working on artificial genetic switches, so Madam, thank you very much again. And I am, and I will be a proud AC project. AC product, as I call myself, Anju Chadha product. Sorry. So be a superstar as you are, and please visit us again. Visit us in Japan soon, please. And thanks for everything. And again, thanks organizer for the opportunity. Thank you very much.
Thank you, Dr. Ganesh.
Shalom, Professor. I, like many others, I'm sure, wish that I could be there on this special occasion that I think commemorates a much longer journey that I was able to be a small part of for a short duration of time. However, I had the pleasure of knowing you as a professor, as a guide, but more than that, I had the privilege of knowing you as a human being and being treated as a friend. The interaction, the conversations, and the banter that we shared, oftentimes, it truly is a special memory and an experience for me to be treated like that by a person of your standing. And when I think of the times that we have had, the times that we shared, even from class, even while we were in lab, while I was working in your lab, I think not of the immediate lessons that are apparent, but I think of those characteristics that are imbibed in me because of my interaction with you. And they are of so much consequence because they are positive and they are now part of my persona and personality. And I'll give you an example, even though this may make this message a little longer, and everyone has a mandate. I'm going to exceed that mandate. When I write my papers, even today for my coursework, I'm writing about history, I'm writing a little bit about political science, international relations. And yet, when I'm working on any of my papers, I'm making progress with the consecutive papers. However, there is an endeavor in each of them where I feel that I have to reach a benchmark where it's good enough that I can send it to Professor Anju. And it's something that I've personally said. It's not something that you have demanded, but it is something that I have said for myself because it makes me better and it comes from you. So each time when I write a paper, when I read it at the end, I think it's moving closer to this obscure benchmark, which I have set for myself, but being inspired by you. And God willing, I can soon send you a paper and share some of my work that I have done. This is just a small anecdote where I want to tell you that it's not lost on me that you've been asking me for something that I have written and I have not sent it to you. It's just that I'm waiting for that day myself. That's good enough to send it to you. And it's a very positive thing. It's not a pressure that bogs me down, but sort of lifts me up to be better and better each day with everything that I do. So I want to thank you, Professor, so much, not for all that you have done, but you keep doing for me, and because of the part that you are playing in my life even today. And no matter where I am, no matter where you are, no matter how long it has been since we have last spoken, that influence will always remain. And my affection for you will always be there, Professor, just as I know that your affection for me will always be there. And I look forward to meeting you. I pray for your good health. Pray for the adventure that lies ahead in the next phase of your life, because superannuation and retirement, these things have connotations of a more, I would say, peaceful life, but I doubt you can relinquish adventure. I doubt people can rest easy with you around. So I think it's just going to be one adventure after the other. It'll be exciting to know what that would look like. And I wish you all the best, all the health, and all the energy and the tenacity for that. God bless those who find themselves around you now. So God bless you, Professor, and a wonderful day and a wonderful time ahead to you.
This is Krishna in Boston. Many congratulations to Professor Anju Chadha, not only for this event, but also for a brilliant and successful career in science. As many of you know, Professor Anju Chadha has been a champion of women in science and also has taken many underprivileged students under her wing and make them successful in the practice of science. She's always fostered engagement between industry and academia, and this stems from the fact that she's worked in industry herself, but also with a keen eye towards her own science being useful and of benefit to society in general and the greater good for everyone. She is never hindered by the powers that be, as many of her students will attest, and she will forever remain the free radical that we have known her to be throughout her career. And so many congratulations once again, and all the best wishes from Boston for both this event and beyond. Thank you.
Thanks a lot, ma'am, for being an amazing professor. You did not only teach us about chemistry or biology, you taught us about life. If there were one thing which I would take away from all your classes, it would be that we need not be politically correct all the time. We have to be fearless, and with conviction we have to stay true to our values and do what we think is right, and not only please people around us. And hand on heart, that is something which will go a long way in helping us stay true to our values. Thanks a lot for that, and wish you an amazing post-retirement life.
Hi ma'am, how are you? You have been always a great inspiration to me. I always admire about your majestic personality. Thank you for your kindness and support to me during my stay in IIT Madras. Wishing you a very happy, healthy, long life. Thanks again. Thank you.
Hi ma'am. When I was sent this message as to whether I can send a video message, I didn't think twice. I had to do it. Ma'am, you are easily one of the best teachers in the department. While you were strict and we were scared of you, we knew that you had our best interests at heart. Ma'am, you pushed us to really put effort in your classes, and it helped me develop and retain an interest in those subjects years after the course. And that's even without me being in that field at this point of time. And outside class, we had a lot of fun. I would enjoy dropping into your office for a casual conversation. I still remember the Harlem Shake and the biryani lunches that we had at lab. In a sense, ma'am, you also not only became a teacher, but you also became a friend for us. I feel really bad for the next generation of biotech students who will never get to learn from you or have a chat with you, ma'am. Hope everything's fine with you, ma'am, during these times. I really wish you all the best in your retired life, ma'am.
Hello everyone. I've known Professor Anju Chadha for a long time now, and first of all I'd like to say that her science, her academics is pure and clean. She will not pass any result unless it's well established. In that sense, she is completely intolerant of sloppy thinking and sloppy experimenting. But on the other hand, in her personal life, she's very tolerant of diversity, of differences of opinion, and she welcomes all of these people with all types of opinions into her room and into her heart. She's especially concerned that the establishment should not have its unfeeling way, and she is fearless in opposing all forms of injustice to her colleagues, to her students, to the general public, and of course to herself. My own introduction to her refreshingly fearless ways of thinking happened nearly 40 years ago when we were contemporary students in IASC. And I saw her passionate activism there from up close. There was one occasion when Prince Charles was visiting the institute, and she publicly and fearlessly spoke her mind to him. That particular occasion is still fondly remembered by us when we meet and talk about her. And spoken of with great respect and admiration, I must say, at all levels. Many of us are now become part of the establishment, but Anju still remains somehow opposed to it and still a part of it. And that's a remarkable quality that she has. And I don't consider myself to be really a part of the establishment hierarchy, so on and so forth. But I know a lot of people who are the top brass, let us say, in science and academia in India. And they all speak of Anju with great respect and affection. So I take this opportunity to wish her all the very best in her future endeavors, that she'll continue to be as fearless as she has always been, and that she'll continue to keep doing good science. I wish her all the very best.
Hello, ma'am. Congratulations on your retirement. First of all, thank you so much for the encouragement or the concern you have shown towards us. And you have always inspired me with your courage, the daring and dashing person you are, and the compassion you have shown towards the students. And for a fact, you are the most admired professor to me and all my friends as well. And I hope you are happy that you are freed from all these annoying students. I really wish you great health, strength, and happiness throughout this retirement. Thank you, ma'am. Happy retirement.
Kindly make a note that those videos which couldn't be played here because of the time constraint will be directly handed over to ma'am. And I thank you all for taking your time out to make those videos.
Sneha, can I take 2 minutes?
Yeah. Yes.
Yeah, I just wanted to take this opportunity to thank Anju. So just would like to thank Anju for being such a good advisor, not only professionally, but also personally. She has always been available at any time whenever I needed her help. I could discuss with her openly all my issues, whether they were personal or professional, and she would always come up with solutions. She will always give me suggestions how to carry forward. And she has been an excellent critique. And I was really fortunate and very lucky to have a person besides me whom because of her excellent critique, I was so confident. Even my students, Narayani, she has always strived for excellence. So I think Narayani learned a lot. I also learned a lot being with her. So thank you, Anju, for spending time. Thank you. And I wish you great health and successful future endeavors, and wishing Ramesh also a healthy and happy times together. Thank you.
Thank you. If anyone wants to share the
Sneha, I would just like to take a couple of minutes. This is Satya.
Okay, sir.
Okay, so Professor Anju, so initially she was a colleague to me. Now I would say she's a good friend of mine, then colleague, then collaborator. So it's very interesting to work with Anju. So apart from science, we had a lot of discussions whenever we meet on various social aspects, especially on women empowerment and gender equality and all that. So it was so nice to talk with her and learn so many things, which generally I don't worry much about it, but she's a very good, inspiring colleague. And with her, we also wrote a joint proposal, and we got a funding from DBT, and we got 3 bioreactors. And our work was a good sign of collaboration between chemistry, biochemistry, and biochemical engineering. So whatever biotransformation she was doing, one of the reactions which involved asymmetric synthesis of S-ethyl 4-chloro-3-hydroxybutane. Okay, so I could not spell it, I copied out on my notebook, and using Candida peroxidase. So actually we scaled up this process from shake flask to I think 20 liters reactor, if I'm right. And we have co-guided a couple of students, an MS student Nivedita and a PhD student Tarjan, who's involved in this bioprocess scale-up this process. And I think your innings is not at over, Anju. So you have a very strong innings now. Okay, so to play in science, I think you won't take rest. I know that. So I will also I suggest you that you should write a book on your journey on in science. Okay, and definitely I think your chapter in that book on ISC Bangalore would be a super hit. And you need to write this book because not only for others, but mainly it will inspire a lot of young women to take up a career in science. Okay, so I wish you and Ramesh a good life. Thank you.
Okay, so this is Karthik. If I can go next, just very briefly.
Yes, sir.
Yeah. So congrats, Anju, on I think it's been a wonderful day. I hope you enjoyed this. So I'll just I have something very quick to say. So Anju and I shared a lot of nice, interesting car rides together. Where we would be talking about absolutely random stuff. And I think one thing that many people, I think some of the students may have remarked, and you can see from the number of young students who turned up today that Anju is actually one of the youngest folks. Right? Don't look at her age on paper, but I think Vignesh is probably digging around for that video. She was the only one who joined when the students did a Harlem Shake in the department. So she's one. So she's like one of the youngest, in mind, of course. And I think that's what counts. And she's inspired a lot of kids all the time. I know that she very enthusiastically does this RSI thing with Professor Mangal Sundar and all the Research Science Initiative in Chennai, where she always has this couple of kids who intern in her lab during the summer and so on. And I think it's difficult to track those guys, but maybe they would have had something really interesting to share about Anju and her lab. I think that's something very unique that I wanted to point out. And that's it. So Anju, have a great time. It was wonderful being your colleague, and I hope you have a lot of fun. And I think Satya has given you something really important to think about. So you better start writing that book.
Okay, so hi, this is Vignesh. So I would like to say a few words as well. So yeah, Karthik has been asking me to find that video. So I have, and I will share that video here. So just to show that the love she had for her students. So that I think that was the greatest thing I had actually observed from her. So I hope you can see the video. So.
Yep, it came on. Is that audio, Vignesh? We don't hear it.
Yeah, I'm dancing. I'm also dancing.
So I hope you guys enjoyed that. So the main thing I saw from her was she always loved to interact with people. So I used to go to the fourth floor to meet with the head regularly, and anytime she has the door open, she would always pop her head out and smile and invite me in. So I have had many times where I have had general conversations with her. I have also reached out to her when one of my students had a difficult time and she was facing some issues, and Professor Anju was the first person I thought of who I could reach out and talk to. So although we hadn't it wasn't that we were friends, I always knew that I could lean on her if I needed something, and she was somebody who I could trust, and that was amazing, considering that it was not that we spent a lot of hours together, but she emanated that quality in her. And the best thing about being with Anju as a colleague is attending meetings. You should always sit next to her. So she made meetings fun. So, and her sense of humor is something I truly enjoy. I loved it. She has a unique sense of humor, and you just have to be there with her in a meeting to have fun. So thank you, Anju,
Not
for all the
everybody gets
memories.
it, but those who get it, yeah.
Thank you, Anju, for all the great times we had. I hope you have a healthy and happy retired life. And what Satya said is absolutely true. We all look forward to that book. I'm sure your challenges and your struggles will be an inspiration for generations to come. Thank you.
So actually, I forgot to tell a couple of things, which nowhere it came about her services to the institute. Actually, she is the first member secretary for Institute Ethics Committee. Okay, so actually, she is the first one to make it very streamlined, and then made all the process very clear, and made that ethics committee popular to many colleagues here. So after her, I took it from her as a member secretary. So it was very, I saw what and all she has done in that one. So she was very meticulous in maintaining the records and all the aspects of the ethics committee. And also she served as a chairman for the Institute C-Cash committee. Okay, I think she has done a wonderful job in that one. And so on behalf of our institute, so I should thank that these are the two major committees that she took over as a responsibility. Did I miss anything, Anju? Institute?
You said more than I was doing.
No, just told what you have done. That's all.
If anyone wants to share experience,
shall I share a few words? I'm Michael. I cherished my interactions with Professor Anju, both in personally as well as on the scientific front. And during our interactions for the about a decade, she gave very good advice in our personal life, as well as how she explains the concept to the kids. Especially, she is very good to explain the school kids in a different way so that the kids can understand. And on the scientific front, I had a collaborative project and collaborative research with Professor Anju, and we combined experimental and computer studies to understand the stereoscopic specific reduction of these specific carbon reductase from her famous enzyme Candida parapsilis with her student Sneka. And I had a good time to interact on these aspects, and on each and every aspect, she was very specific to be very clear to before we move on to the next aspect. That's what I liked very much with her, and I enjoyed our interactions with her, and I wish her a happy and healthy and safe life. Thank you.
any more experiences if people
Can
want
I
to share?
say something? This is Manoj here.
Yes, sir. Yes, sir.
Hi, Anju.
Hi, Manoj.
I can tell you when I first met you, okay? July 1st, 2004, 11:10 a.m.
Oh my
I
God.
joined the institute, and I met Guhan, who was the HOD then. I reported to him, and then he took me out, and the first person we met in the corridor was you, okay? And of course, this IASC affinity is there, but then, you know, few hours was enough. You know, I sort of had a natural affinity for somebody like you. My social circle is very small, professionally and personally, but there are very few people in that circle, and definitely you are one of them. Okay, it has always been like that, and I'm sure it's going to be like that. I definitely admire, you know, the thoughts that you have. I share so much of that with you. Okay, I have almost the same thoughts, but I have, you know, maybe 1,000th of the guts to state that. Okay, that's something, you know, I always admire, and I think that's what this natural affinity is. Our line of thought regarding many things, whether it is religion or, you know, whatever random things, we share a lot of similarities. But, you know, I just keep mum about that, whereas I always think I should someday in my life I should get to some level of, you know, confidence where I can state it like you can do. Okay, I appreciate all this, all the association we had. We will remain so, Anju. So formally, let me again wish you all the best. No, we will continue to remain like this, fine, as friends. Okay, and yeah, you should remain as it is, meaning when as you are now. Okay, absolutely no change, right? And yeah, we will remain, we will be together. We will share these same things. Yeah. Thank you.
Thank
So
you.
at this point, just one comment. So as a department, we should thank Anju, Manoj, and Gopal because they put a lot of efforts in getting DST FIS grant, so which most of us are using the CD and what is that one? DSC. Okay, thank you.
I thank all those who have put in their wonderful memories here. A leader is one who lights the path to the impossible and takes those who follow to great heights. It is an injustice to proceed to the conclusion without calling upon our wings of light, our pillar of strength, and our constant source of motivation. We cordially invite our adored Professor Anju Chadha to share her experience and memories with us. Ma'am?
Yeah. Can you hear me?
Yes.
We can hear you.
So, all colleagues of the Department of Biotechnology, dear Professor KKB, and of course, my very dear students, without whom this journey is absolutely meaningless and impossible. You can be the best teacher, but you must have somebody to teach. And of course, I have been very lucky that I've had a collection of very important and wonderful people who have lent themselves to get molded and turn out to be such fine human beings. See, the seed has to be there for it to develop, and all of them, each one of them is a gem. Whether they were undergraduates, postgraduates who came to me, or project students who carry with them nice memories of time spent together. I thank you from the bottom of my heart. Professor KKB, you have been a wonderful associate, and I have always cherished my memories with you, whether it's work related or otherwise. I think this has gone a long way in trying to define what science is about in India and what one can do. Long years ago at IASC, Professor Balram, my other teacher, and someone I could get along with and talk to, had told me when I got back from the US, Anju, in India, you can't always do what you want to do, so you do what is doable. And I just want to tell you at this point that if you think that I have too many interests, the underlying theme has always been the enzymes, whether they be applied in biosensors or in biodiesel or translational work, it's always enzymes which has taken my interest away, right from their understanding basics to the application. So even though it might seem diverse, it's somewhere connected with this important lesson which I had seemed to internalize so dearly. And I think it makes sense, because I think if you have a scientific temperament and you question, it's inevitable that you want to try and solve the problems as you go along, regardless of this terribly theoretical issue about only wanting to understand this much and not more, and therefore have blinkers. That's never been my cup of tea. It has to be understood, it has to be clear, and it has to move on. So I have benefited a lot from people like Professor KKB on the way, and without hesitation, he makes industrial work and application-oriented work sound equally good and interesting to basic sciences. It need not always be a very basic theoretical question. It can be equally interesting if you're trying to house a pre-filter in a bioprocess plant. It has to be done properly. You have to understand it. So this has been a great motivating factor for me. And I think the students also love it when you tell them, trust them, they rise to the occasion. Tell them that the lab is meant for you, not the other way around. They do it and they learn. And that is what is required today. Much as whatever else people might say, the leaders might say, the policymakers might say, the fact of the matter is that Indian young people in India need hands-on training in a lot of things. Without that, all this sloganizing about Atmanirbhar and all can go fly a kite. So to the colleagues, of course, trust people. Doesn't matter if it breaks, maybe once, you can always repair it, you can always get a new one, replace it, but please give the youngsters a chance. They are really, really equipped for this and they will carry it through. And I'm deeply grateful to Professor Satya, Vignesh, Guhan, Manoj, Michael, all the colleagues in the department, Smita, who I have associated with quite closely, and I don't see Mahalingam here, but Mahalingam on the same floor has been helping once in a while. I remember going to him and telling him, I don't have any funds to buy a -20, my -20 is conked off. And he just said, here, there's one which has just come for the new DST project, you can have it. Yeah, this is a nice thing. This is how it should be. And I have done the same whenever I could spare it, whenever I have been approached. They are meant for us. We are not meant for these things, you see. The more you use instruments, the better they remain. That is really a truth. And KKB believed in that. I saw him functioning in chemistry. People from all over would come to use instruments. They are meant for that. And more people use it, the better it is. So I have had a great time. Vignesh, I really want to thank you for the lovely dance, I wish we could have done that longer. The Harlem Shake. It was very nice. It was a lot of fun. And of course, age no bar, of course, it's just a number. I agree with that. So I'm glad you appreciate my wacko sense of humor whenever it wasn't too sharp for others. So big thank you to the department. I also want to thank the staff in the office, Sita Lakshmi and Sai and the others who made it so much better to function there. The housekeeping staff, the women there who were relentlessly keeping the place clean and in order. The whole lot. I mean, IIT staff from top to bottom. I go to ICSR and people are there ready to help. So a big thank you to each one. And of course, my chemistry colleagues, I cannot forget Professor I.P. Singh, SSR, and other young colleagues from chemistry who have always been nice to interact and of course learn from. So deeply grateful to everybody. I'm sorry if I've missed anyone, but I carry you and your memories in my heart. Ganesh, I have registered what you have said. Yes, lucky bug you are. So thanks again and all the very best. Let's hope we meet sometime in the future minus all this pandemic nonsense. Goodbye and good luck.
Thank you, ma'am. Thank you for sharing your experience with us. As we draw towards the conclusion of this splendid assembly, I would be diligent if I did not mention the people who made this mini-symposium possible. To do that, I invite Dr. Saravanan, a former student of Professor Anju Chadha and currently a faculty at Chemistry, University of Hyderabad, to deliver the vote of thanks.
Is it audible, my voice?
Yes, we can hear you.
Yeah, thank you. First of all, good afternoon, everyone. It's indeed a great pleasure and honor to propose the vote of thanks on this occasion. First and foremost, my heartfelt thanks to our organizers, Professor Satyanarayan Kumadi and Dr. Viknesh Muthu Vijayan, who, despite their busy schedule, have found time to arrange this wonderful event, providing a valuable suggestion and support to ensure this event become a grand success. And I extend my special thanks to Professor Gon Jayaraman, head of the department, and other faculty members and non-teaching staffs of the Department of Biotechnology for their support and valuable time to this event. I express my sincere gratitude to Professor KKB, Dr. George Thomas, Dr. Deepti Sarin, and Dr. Santosh Kumar Pati for readily accepting our invitation for sharing their research experience in this wonderful event, especially for enlightening us all with their knowledge today. Also, I thank all the faculty members and students from IITM and other institutions for attending this event, also for sharing their memorable experience with me. I thank Dr. K. Saravanan, Dr. Vinay and Muthu for collecting all these wonderful videos and playing it here nicely. And I thank Dr. Kabilan and Leena for e-flyer. Also, I thank Dr. Sneha and Jerina for comparing this function. Last but not least, I thank all the participants for your cooperation and attention in making this event a resounding success. Once again, thank you all for your attention.
Thank you, Dr. T. Saravanan. Ending with a quote which says, "Sometimes you will never know the value of a moment until it becomes a memory." With that very thought, let's treasure this memory for a lifetime. I thank you everyone for being a part of it. Thanks a lot.
Thank you.
Thank you, ma'am.